Hi guys, did everybody get a notice that this room's being recorded? Awesome. All right, if you'll give me just a couple minutes or just a couple seconds, I can get this set up. All right, can everybody see this being shared? Can I get a thumbs up or a thumbs down? Okay, awesome. So yeah, everything that they said I'd like to relay to you guys. I'm sure I kind of parsed through this. There's going to be some really cool research that we get to hear about. Um, right now I'm stalling for time. We're going to start in 3 minutes. Um, Yeah, I just like to once again commend all of you on this rather intensive summer of research that you've done. Um, yeah, and I look forward to it. So I guess actually, if it's okay, we can start a bit early with Shreyas. I hope I say that right. If you're not ready, you still want a minute or not or 2 minutes, feel free to let me know. Um, but yeah, this is going to be our first presenter, and I'll turn it over to you.
Good morning, everyone. My name is Shreyas Damera. I'm from Paradise Valley High School, and alongside my mentor, Miss Faley from Northeastern University, we conducted a systematic literature review titled Optimizing Thermal Incineration for PFAS Destruction, a review of operating parameters and byproduct formation. Per and polyfluoroalkyl substances, also known as PFAS, is a synthetic chemical found in the environment known for their persistence and resistance to degradation. This chemical is linked to serious health effects such as immune dysfunction, thyroid, and cancer. Incineration, which is a mass destruction technique used to destroy PFAS, although it's the most promising, if it's not tightly controlled, it can lead to fluorinated byproducts and not result in the mineralization of PFAS. The goal of the systematic literature review was to identify critical operating conditions, which would result in maximum destruction efficiency while also minimizing emissions and residues. We followed PRISMA 2020 guidelines, and throughout our screening process, we focused on papers generally around PFAS and thermal incineration and around the four levers of operation: temperature, residence time, oxygen concentration, and waste composition. And then we mapped these papers to three incinerators: hazardous waste incinerators, rotary kilns, and waste energy slash sludge incinerators. Of the first four operating parameters, temperature, many experiments concluded that 1000 degrees Celsius is needed for greater than 99.99% destruction, especially for stable compounds such as perfluorooctane sulfonic acid. Below this, if the temperature drops for even a short moment, then persistent byproducts such as sulfonic tetrafluoride are commonly detected. Then the interval between 1100 to 1200 degrees Celsius is when the reaction accelerates rapidly. This is usually the sweet spot for many incinerators. The second operating parameter is residence time. Longer residence time correlates with higher destruction efficiency. Usually, 2 seconds of residence time enables complete oxidation, and underneath this results in partially oxidized byproducts. The PFAS injection timing also matters here as well. Late injection of PFAS into the high temperature zone results in byproducts leaving in effluent gases. This is relevant to both short chain and long chain PFAS, but for long chain PFAS, it differs. It requires both extended residence time and elevated temperature. This is because incineration happens incrementally to the chain, and since long chain is longer than short chain, it requires extended times. Next is oxygen concentration. Oxygen-rich environments result in full combustion and fewer residuals, while oxygen-deficient environments result in byproducts such as volatile organofluorine species, acyl fluorides, and halogenated byproducts. This essentially means that low oxygen results in increased environmental risk via toxic transformational pathways. The last operating parameter is waste composition. This essentially means that what's being incinerated affects PFAS destruction and byproduct formation. For example, sludge is easily degradable, so it results in 99.9 destruction, and PFAS is often trapped in ash. Then, for plastics, since they're harder to degrade, it results in more toxic byproducts and low oxygen. Furthermore, in ash residues, it can retain leachable PFAS, so even though sludge results in PFAS often trapped in ash, it needs post-combustion treatment plants. So essentially, treatment needs should be adjusted conditions to waste type. Next, in byproduct outcomes across conditions, the most important information found is that there's a need for oxidative conditions to avoid stable intermediates. For example, low oxygen slash temperature results in VOS slash halogenated PFAS. Next, for destruction efficiency, high destruction efficiency is achieved in optimized systems, but there is operational limits in full-scale incinerators, and it can mask residual emissions. Lastly, for chemical speciation, monitoring is extremely important since it can detect byproducts missed by DE metrics alone. So these results can be mapped to three incinerators: hazardous waste incinerators, rotary kilns, and waste to energy slash sludge incinerators. Out of these three incinerators, hazardous waste incinerators are the best since they result in more than 30% destruction efficiency than waste to energy slash sludge incinerators, more than 5% destruction efficiency than rotary kilns. Although they do require higher operating time and higher residence time, the destruction efficiency outweighs these costs. So, in conclusion, PFAS can be effectively destroyed by incinerators, but only with carefully controlled operating conditions. These operating conditions being temperature, residence time, oxygen concentration, and waste composition. With robust treatment safeguards to prevent secondary contamination, incineration remains as the most feasible large-scale method for PFAS destruction. I'd like to thank my mentor, Ms. Faley, for her support and guidance throughout this entire systematic review process, and Open Bio SRI staff for conducting the summer research program. Thank you.
Awesome. I think that was a great first start. Um, Looks like next we will have, let's see this, Stella with, oh, hold on, I'm trying to get rid of my Zoom thing. I'm not gonna try to pronounce it. So I'll let you introduce us.
All right. Hi, my name is Stella, and I'm a rising senior at Harker. And my mentor was Miranda Sramoski from the University of Maryland. And so basically, I did a non-systematic review on oligonucleotide-based therapeutics for cancer therapy, specifically ASOs, siRNA, and aptamers. So I think many of us know already that cancer is one of the leading causes of death, despite decades of developing treatments to improve outcomes. And the main reason for this is because of off-target toxicity. So treatments such as chemotherapy and surgery are often not very specific. Where chemotherapy, it often kills both cancer cells and healthy cells, and this can lead to a lot of adverse side effects. As for surgery, while it may be more targeted, it's very invasive and only works for solid tumors. And so this is where oligonucleotide-based therapeutics come in. So oligonucleotides are short strands of nucleic acids, and they can help address the problem of off-target toxicity. So the main three that I focused on for this review were antisense oligonucleotides, or ASOs, and these bind to mRNA, thus blocking translation. And so proteins necessary for cellular functions can't get made. And then I also looked into small interfering RNA, or siRNA, and these incorporate themselves into a protein complex called the RNA-induced silencing complex. And this complex cleaves the mRNA that is complementary to the siRNA. And then finally, I looked into aptamers. So these are oligonucleotides that fold into 3D shapes, and similar to like normal drugs or antibodies, they can bind to or inhibit specific proteins. And so I used 5 papers for each modality. So I'm going to dive a little bit into my results. So ASOs are highly specific, and this is because they only bind to their complementary mRNA. And a lot of studies show that combination therapy was very promising. So they combined like ASOs with chemo, and that showed positive outcomes. So researchers should optimize like dosing and finding which combinations work best. As for siRNA, interestingly, many studies involve like nanoparticle delivery systems, and this is probably to overcome the RNA's negative charge. And because of these delivery systems, they're able to pass through the blood-brain barrier pretty well, or other physiological barriers. However, researchers still need to focus on enhancing stability and identifying new targets. And then finally, for aptamers, these are able to access sterically hindered targets pretty well due to their high binding affinity. And so this allows for more drug accumulation at the target site, and this can minimize off-target effects. so addressing the limitations of like chemotherapy. And again, these are also seen to be effective in combination treatments similar to the ASOs. And so aptamers are pretty safe and effective. And so ultimately, future efforts should prioritize the further development of these oligonucleotide-based therapeutics for more personalized cancer treatments. And yeah, that's pretty much it. I'd like to thank my mentor, as well as SRI for providing the resources and opportunity to conduct this research. Thank you.
Awesome. Yeah, I think we're off to a great start. I mean, I'm sure I'm probably going to say that about most of you guys. But yeah, great research, great research. Learning a lot. Okay, next we have Sucha Reale talking about cancer again, the G12C inhibitor efficacy. Hopefully, I'm introducing that correctly.
Hello, I'm Suchariali, a rising senior at Lafayette High School in St. Louis, Missouri. Our project focused on the KRAS G12C mutation, long considered one of the most undruggable targets in cancer biology. KRAS is a small GTPase, and you can think of it kind of like a light switch. In its GTP-bound state, it's on, constantly sending signals that tell the cell to grow, divide, and survive. In its GDP-bound state, it's off. In healthy cells, KRAS flips on and off in tight coordination, but mutations like G12C break that switch. It gets stuck in the on position, driving continuous tumor growth. You can see that in the MAP kinase pathway and the mTOR axis in the top middle in Figure 1. This mutation is common in non-small cell lung cancer, less so in colorectal cancer, and even rarer in pancreatic ductal adenocarcinoma, which you can see in Figure 2 on the bottom row. For decades, the problem was structural. KRAS had no deep binding pockets and gripped its guanine nucleotides with picomolar affinity, making it nearly impossible for a drug to wedge in. That changed in 2013 with the discovery of a switch II pocket, an opening that allowed covalent inhibitors like sotorasib and adagrasib to lock KRAS G12C in its inactive GDP-bound state. In preclinical xenografts, these drugs erased tumors, but when they reached patients, the picture was more complicated. Some cancer types responded dramatically, others far less so. That's where our work comes in. We conducted a PRISMA 2020 systematic review and meta-analysis to create a real-time clinical map of where these inhibitors stand and how to get the most out of them for patients. We screened every trial since 2013, over 826 manuscripts, finally landing on 18 manuscripts for 9 unique studies. And then we stratified by cancer type and agent. You can see that in the bottom left in Figure 3. Let's start off with non-small cell lung cancer. This is KRAS G12C inhibition's biggest success story. Across agents, these drugs consistently shrink tumors and extend progression-free survival beyond what standard chemotherapy can offer. Agents like IBI 351 and glycerasib are showing especially favorable efficacy and tolerability in early phase studies, and the phase 3 CodeBreak 200 trial proved sotorasib outperforms docetaxel in the second-line setting. So why the consistency? Lung cancer tumors tend to be heavily addicted to KRAS, as it's their main power source, and they have fewer bypass pathways to reroute signaling when KRAS is shut off. In this context, monotherapy really works. Colorectal cancer tells a different story. Here, monotherapy works briefly, but epidermal growth factor, or EGFR, rapidly reactivates the MAP kinase pathway, restoring tumor growth. The fix? Vertical pathway blockade, hitting KRAS and EGFR at the same time. For example, when cetuximab is added, especially to sotorasib, the tumor's escape routes close, response improves dramatically, and progression slows. Specifically, the treatment increased response rates up to 62.5% and extended survival up to 8.1 months. That's just not a statistical bump, but it's a mechanistic win because it argues for frontline combination therapies. Then there's pancreatic cancer, the toughest cancer to crack. Even with KRAS shut off, the tumor keeps growing, thanks to its dense stroma, poor drug penetration, and backup survival programs driven by co-mutations in TP53, SMAD4, and CDK92A. Here, single agent KRAS inhibition is like cutting one wire in a bomb with 10 redundancies. It's just not enough. But isolated responses suggest that if we target the stroma, alter the microenvironment, and hit the KRAS simultaneously, we might finally be able to make progress. Across all cancers, these drugs are well tolerated for the most part. Serious side effects are uncommon but manageable, even in combination regimens, meaning there's room to be aggressive with combination strategies without sacrificing safety. Grade 3 and 4 adverse effects occurred in 9 to 34% of patients. So why does this matter? Because for the first time in history, we've been able to drug KRAS in patients, a target that drives nearly 1 in 4 cancers. These therapies are already changing the standard of care for lung cancer, they have a clear path forward in colorectal cancer, and they're showing us exactly what barriers we need to break in pancreatic cancer. This is precision oncology in action, and the patients who will benefit are already in the clinics today. I'd like to thank my mentor, Dr. Andrew Evans, for his guidance, Dr. Gregory Petsko for his invaluable feedback on my project and manuscript, and the NIH for funding. Thank you.
Wow, I think that was a pretty strong presentation and comprehensive review. Um, Looks like next we'll have Vahedi with environmental endocrine disruptors.
Hi, my name is Vaidhi Hinduja, and my scoping review was conducted with guidance from my mentor, Maisie Radcliffe from the University of Florida, and is titled Environmental Endocrine Disruptors in Air and Water and Their Association with Breast Cancer Risk, a scoping review. Breast cancer is one of the most prevalent malignancies in the world, affecting 1 in 8 women over the course of their lifetime. While BRCA1 and BRCA2 mutations dominate public discourse, genetic predisposition explains only a fraction of cases. Increasingly, the scientific community is turning towards environmental exposures as potential modifiable drivers of carcinogenesis. EDCs are exogenous compounds capable of agonizing or antagonizing hormonal receptors, most notably the estrogen receptor, thereby disturbing estrogen homeostasis. Examples include benzene, particulate matter PM2.5, PFAS or forever chemicals, DDT residues, and cadmium, just to name a few. Now, airborne EDCs, which are emitted from petrochemical facilities, high traffic areas, and incineration sites, can be inhaled into pulmonary alveoli, transfer into circulation, and then interact with breast epithelial cells. For example, multiple cohort studies reported significant associations between long-term PM2.5 exposure and breast cancer risk, with hazard ratios ranging from 1.11 to 1.85 per 10 micrograms per cubic meter increase. PM2.5 can carry fat-soluble chemicals into the body, trigger inflammation, which can cause oxidative stress and disrupt estrogen metabolism. Another example is benzene, which has been linked to DNA breaks, the production of harmful reactive oxygen molecules, and changes in chromosomes. PAHs derived from incomplete combustion can form bulky DNA adducts and epigenetically silence tumor suppressor genes such as BRCA1, particularly in individuals carrying the CYP1A1 or CYP1B1 polymorphisms. Now, waterborne exposures present a different but equally concerning route. PFAS, often called forever chemicals, persist in groundwater for decades and bioaccumulate in adipose tissue. Now, while meta-analyses show modest relative risks, around 1.08 for PFOA, their environmental persistence means long, chronic, low-dose exposure and is virtually inescapable in contaminated regions. DDE, a fat-soluble metabolite of DDT, has been shown to double breast cancer risk in certain populations, with elevated odd ratios for OP-DDT exceeding 2.2 in agricultural zones with historical pesticide use. Another example is cadmium, which is often introduced into water systems via industrial runoff and can bind to estrogen receptors and activate transcriptional programs similar to estradiol. Urinary cadmium concentrations in the highest exposure quartile have been associated with up to a 3.74-fold increase in breast cancer risk, alongside evidence of metabolic mediation through insulin resistance. Now, my scoping review synthesized 52 peer-reviewed articles published between 2010 and 2025. While findings vary due to different study designs and exposure assessment methods, the overwhelming amount of evidence points towards an environmentally mediated pathway for estrogen receptor positive breast carcinogenesis. Beyond individual pollutants, an important theme also emerged: cumulative and synergistic effects. Mixtures of persistent, bioaccumulative, and toxic chemicals often demonstrated stronger association with breast cancer than any single compound alone, suggesting that real-world exposures cannot be fully understood in isolation. Now, this matters because environmental risk is not evenly distributed. Communities near petrochemical plants, hazardous waste sites, and areas with historical pesticide use, often those with lower socioeconomic statuses, are disproportionately burdened. Public health frameworks must evolve to integrate environmental monitoring into cancer prevention strategies, prioritize biomarker-based exposure assessments, and address vulnerable development windows such as prenatal and early life exposure. If breast cancer prevention is to move beyond genetics, we must confront the reality that clean air and clean water are not just privileges, they are prerequisites for health. I would like to thank my mentor and the Open Bio staff for the opportunity to conduct this scoping review. Thank you.
Awesome. I think that was pretty well. I'm probably gonna say that about everybody, but yeah, I really liked your nuance that you had, as well as your scoping review of all the different pollutants. All right. Next we have a video from somebody that could not make it today. Um, it looked like I believe it was about 7 minutes. So I don't make the decision on that. I guess we're gonna watch it for the 7 minutes. Hopefully, this will go good. You guys can't hear it?
We
Okay.
can.
All right. So we're gonna move on from that. I can ask Sahil because he set that up. Um, so yeah, it looks like next then we will have, and if you want to take a little break to get ready, that's okay too for this sudden jump. But we have Eileen. Hopefully, I'm saying everybody's names right. Between video game addiction and loneliness and social disconnection, I think this is a very impactful topic today.
Hi, my name is Eileen Putla, and my mentor was Madeline Pelgrim. And my research explores how video game addiction impacts adolescents' mental health, specifically loneliness and social disconnection. Gaming is no longer a solitary pastime. It's immersive, social, and extremely popular among youth. While moderate gaming can be healthy, excessive gaming raises concerns about whether it replaces rather than supplements real world interaction. In 2018, around 8.5% of adolescents and children were estimated to have a gaming addiction. This number has since increased due to significant changes in gaming habits since the COVID-19 pandemic. My goal was to examine the psychological factors behind gaming addiction and its effects on adolescent social well-being by conducting a literature review. A key concept in this discussion was internet gaming disorder, the clinical term used to describe a gaming addiction. Internet gaming disorder is defined as a compulsive need to play video games, often at the expense of other life responsibilities. There are 9 clinical criteria for an internet gaming disorder, including preoccupation with games, withdrawal when you can't play, loss of interest in other hobbies, lying about gaming, and continuing to play despite clear problems. Unlike casual gaming, internet gaming disorder is marked by severity and significant impact, often linked to depression, anxiety, low self-esteem, and social discomfort. Many use gaming as a way to escape real-life stresses, creating a reinforced cycle of avoidance. I also looked at some theoretical framework to understand the psychology behind a gaming addiction. Some notable models surrounding this topic were Davis's cognitive behavioral model and Kaplan's adaptation on that model. Both explained excessive gaming as a product of psychological vulnerabilities and maladaptive cognitions like anxiety or low self-esteem that make online environments feel safer and more rewarding than real-life interactions. In Davis's view, this reliance on gaming for mood regulation and social validation can evolve into specific pathological internet use, marked by compulsive behavior and withdrawal from offline relationships. Kaplan adds that a preference for online social interaction and use of gaming for mood regulation impairs self-control, leading to cognitive preoccupation, compulsive use, and negative outcomes such as academic decline, poor relationships, and increased isolation. Together, these models show how excessive gaming can shift from a coping mechanism into a self-reinforcing cycle that deepens loneliness and social disconnection. What makes this cycle particularly harmful for adolescents is that it disrupts a critical stage of development. Adolescence is when individuals learn how to build lasting relationships, navigate social dynamics, and develop their identities in real-world environments. When excessive gaming interferes with these processes, it can stunt emotional growth and limit opportunities for healthy interpersonal connection. Models like Davis's and Kaplan's highlight the role of maladaptive thoughts, poor coping strategies, and preference for online interaction in the development of the disorder. While not universally recognized in healthcare, the World Health Organization officially classified gaming disorder in 2018, prompting treatments like cognitive behavioral therapy and family interventions. While gaming does offer a sense of community and temporary emotional relief, it can also become a dysfunctional coping mechanism when misused among individuals with existing mental health issues. Youth with gaming addiction symptoms often experience greater loneliness, lower school engagement, and reduced life satisfaction. For vulnerable adolescents, gaming can shift from recreation to a harmful substitute for social development. Recognizing unhealthy patterns and promoting balanced digital habits is essential to support online and offline growth. This topic is highly nuanced as gaming is not inherently harmful. Further research and awareness can help to distinguish between harmless recreational gaming and dependency, ensuring that young people are supported in developing healthy, balanced social interactions across digital and real-world environments. I'd like to thank my mentor, Madeline Pelgrim, for her invaluable guidance and support throughout this process, and the SRI staff for this incredible opportunity to conduct my own research. Thank you.
Awesome. I think you explained it fairly or pretty well. And I'm really liking how everybody has such nuanced positions in their research and are willing to admit that. Great job. All right, so next we have John, uh, and he is working on hemoglobin or sickle cell disease. We'll hear more from him.
Hello, my name is Maxwell John Luther, and I'm presenting on predicting HbF score for sickle cell disease. Sickle cell disease is a debilitating genetic disorder influenced by numerous genetic modifiers. These genetic modifiers contribute largely to the disparities in severity among sickle cell patients. For instance, if you take a particular population, the severity of sickle cell disease among those populations are quite different. And the reason is tied to some of the genetic modifiers that contribute to this severity. So in our research with my mentor, Daniel Araujo, we were able to use statistics from the genome-wide associations and candidate gene analysis to review some of the statistics that have been labeled over there. And we found out that there are quite few number of this genetic disorder that affects sickle cell disease, particularly when you consider HbF score, because the HbF, which is the hemoglobin, the fetal hemoglobin, is a standard process that serves as a protective trait for labeling or leveling the severity of sickle cell. So in doing so, we wanted to find out how these genetic disorders, these genetic variants are able to predict how the HbF scores are. And if you look at our table one, these were the variants that we discovered from the GeneWAS catalog and the RIX allele. These are the alleles that affect, that contribute or bring about the severity. And their beta values, which is over there, tells us the percentage of how these genetic variants or genetic mutants contribute to the HbF levels, which is the hemoglobin fetal level. And in our table, you realize that the first variant over there increases the HbF level by 0.58, which is quite the highest number we achieved from our GWAS catalog. And assessing superpopulations in our study, we have Africa, America, East and Europe, and Southeast Asia. So in bringing about testing for the HbF scores, this was what we did. We grouped each individual's genotype that is related from the GWAS catalog, and we also got information from the variants, which is their beta values, and we multiply them. So this genetic data or the genotype that we got was in was transmitted into dosages and were multiplied by the beta values. And we realized that in the population that we have here, the severity that we considered that it varies was quite significant for people in Africa population. If you look at figure 2, you will see that the individuals that are in each of the populations in Africa have quite higher severity than any other population. And well, this is similar to the Europe as well. And we wanted to find out what these differences could be. So if you check our figure 3, we group these these populations into superpopulations in Africa, Europe, America, Southeast Asia, and East Asia. And we tested for their differences, which is their main difference in the HbF levels. And we realized that for Africa and other populations, except for Europe, the HbF differences was quite significant. But for Africa and Europe, there was no significant value, which in our study implies that Africa and Europe are the ones more susceptible to having the severity of sickle cell disease. So in all, we we did our data with a quite small number of the variants that we discovered, and we are hoping that future researchers will be able to explore more of these genetic variants so that it will be quite feasible for us to determine personalized medicine for sickle cell disease. Because in sickle cell disease, due to the changes in their complications, we cannot actually best fit a medication for an individual. So what we did was that based on our predictions and our model, we were able to come out with a conclusion, predict that Africa and Europe would likely benefit with hydroxyurea. Hydroxyurea increases the level of HbF. So if Africa and Europe have a lesser HbF levels as seen in our data, they would quite benefit from this treatment. And also future researchers to also explore other means of getting personalized medicine for these sickle cell patients. And our studies, we took it was the resources from Chicago Research Computing Sector and my mentor as well, and also the funding from Harvard Open Bio for making it a great success. Thank you.
Awesome. I really liked at the end how you had the uh you proposed a solution for those different populations. Yeah, I think that showed a lot of nuance. A common theme of this breakout room is that everybody has a lot of nuance. And I really commend you all for that. Next up, we have Tosha Patel um talking about crustacean mediated disease dynamics. Take it over.
Hi, my name is Tosha Patel, and I'm a rising junior at Blue Valley Southwest High School in Kansas. When you think of disease transmission, do crustaceans such as lobsters and crabs come to mind? Well, this review dives into the important value of disease dynamics in such creatures. Disease dynamics in animal populations are fundamentally driven by social behaviors like aggregation, which influences both the transmission of pathogens and susceptibility of hosts. In crustaceans, a group known for the diverse ecological roles and social structures, these dynamics are especially pronounced. This study provides a systematic review of crustacean disease dynamics, including the behavioral and environmental drivers as well. Aggregation is a widespread social strategy in the form of group living. In simpler terms, it's when animals group together to survive external pressures like predators, weather, and more. We wanted to dive into how disease spread was influenced in crustaceans that aggregate. This study is especially important in understanding the behavioral and ecological structure needed to advance the management of disease in ecosystems, as well as provide the basic disease transmission framework applying to many other species. Additionally, it can be beneficial to apply to human epidemiology and host pathogen relationships. To explore this, we conducted a systematic review using the PRISMA method. We searched databases like Google Scholar, JSTOR, and PubMed for relevant studies. These are summarized in the PRISMA flow diagram in the methods column. We made sure that these studies were adapted from various years to encounter for the newer as well as the past scientific developments to have the best range of records. The range of the years of the records are included are shown on the table 1 in the methods column underneath the Prisma flow diagram. We split our findings into 4 categories. First, for disease susceptibility, crustaceans as parasites. Crustaceans can also act as parasites of the disease. A study conducted in 2 records expressed that a higher host aggregation results in a dilution effect of the crustacean parasite, meaning that individual infection risk was lowered, while the infection success was unaffected. If you look at Figure 2, it analyzes the relationship between the high host densities of Atlantic salmon diluting and reducing the parasite sea lice, a smaller individual infection risk of the salmon. Second, for disease transmission crustaceans as hosts of the disease, studies show that shell disease caused by Aquamarina SPP and white leg disease in decapod crustaceans are primarily caused by environmental factors. And a major trigger to these diseases were shell abrasions. However, there isn't enough information to conclude that aggregation is a prime driver of increased abrasions, even though considering the increased physical contact that aggregation has. For behavioral mechanisms in relation to disease avoidance, 3 major cues were used. 1, visual, 2, chemical, and 3, auditory and mechanosensory. These cues differed based on the environment, such as the cues differed in aquatic versus terrestrial environments. That ties into the 4th and final key finding, environmental influence on crustaceans' disease dynamics. A study showed that Caribbean spiny lobsters in artificial shelters called casitas with various vegetation amounts in the environment were actually more crowded in zones with higher diseased individuals, whereas in the lab setting, these lobsters highly avoided diseased individuals. This comes to show that lobsters' preference on the shelter was highly influenced by environmental factors, such as increased biomass to protect them from sunlight and predators, which was not present in the lab settings. In conclusion, after reviewing these topics and knowledge in the field of disease dynamics of crustaceans suggests that aggregation and environmental factors and behaviors are drivers of disease dynamics. Additionally, aggregation still has the potential to increase the speed of disease spread, inducing a epidemiological like effect. However, it lowers the individual infection risk. It may be beneficial for future studies to focus on factors that increase abrasions and leg abrasions and factors that account for parasite outbreaks to have an enlightened view of the study of this field. I would like to thank you all for listening, and I would like to thank the SRI staff for giving me this opportunity to conduct this research, my family, and my mentor, Madison Karam, for her work in this project. Thank you.
Wow, I think that was a very interesting topic. Definitely something I've not researched before, so thank you for that. Um, all right, so the next person that we have is Sarayu, and it looks like we are looking at drivers of wolf livestock conflict. Um, I'll turn it over to you.
Hi, my name is Sarah Valerapalli, and I'm a rising senior at the Edison Academy. For this project, I was mentored by Lara Menchak from Oregon State University. And in this review, I examined drivers of wolf-livestock conflict to inform management and future research. Since the 1990s, the reintroduction and natural recolonization of gray wolves in the northwestern United States have led to a steady population increase. However, this recovery has brought wolves closer to rural livestock operations, resulting in escalating wolf-livestock conflict. As a result, livestock depredation by wolves has become a significant source of economic loss for producers. Many studies have focused on the seasonal and ecological factors that influence depredation patterns. The purpose of this literature review was to 1. Summarize the factors influencing wolf depredations of livestock, and 2. Develop a list of covariates for future analyses. By identifying key covariates associated with depredation risk, this review aims to support more informed management decisions and contribute to the development of predictive models for future conflict prevention. We began with a Google Scholar search of wolf and depredation and selected the papers that focus on wolf-livestock conflict across the United States. We then created a spreadsheet to track the variables of interest, hereafter covariates, mentioned in each paper. After identifying key factors related to wolf depredation, we grouped them into 4 broad categories: wolf pack, climate and environmental, livestock operation, and anthropogenic characteristics. Finally, we recorded whether each covariate was associated with an increase or a decrease in livestock losses due to wolf depredation. And here are our results. The wolf pack category includes biological and behavioral traits of wolf packs that may influence the likelihood of livestock depredation. Range expansion, including colonization of new areas, increased depredation risk. Previous depredation also contributed to the increase, meaning that farms experiencing depredations are more likely to experience future depredations. Pack size is also a factor, with larger packs more likely to exhibit recurring depredations. Lethal control of wolves, on the other hand, was found to have conflicting impacts on future depredations, with some studies showing increased or little impact and others showing that targeted partial pack removal can reduce future depredation risk. In the climate and environmental category, we talked about climate or environmental patterns that can affect wolf movements, hunting success, and interactions with livestock. Greater vegetation cover may increase depredation risk, particularly during denning season. Distance from forest edge was also found to have no significant effect. Snow accumulation may increase wild prey vulnerability, reducing livestock depredation. Alternatively, it may draw wild prey near ranches, so snow accumulation was one of the more equivocal factors. Land cover types, such as forest, pasture, and cropland, may predict depredation risk, but the strength and direction of these relationships can vary depending on local grazing practices and habitat configuration. Next, the livestock operation characteristics category includes operational and management practices that influence livestock vulnerability to wolf predation. Distance to wolf dens may increase depredation risks. And calving site, calving duration, or start date were found to have no significant effect on depredation risk. Breeder type of cattle were also similarly found to have no significant effect on the risk of cattle depredation. Larger pastures, pastures with more cattle, and pastures farther from human residents are more likely to experience depredations. And relative abundance of cattle within wolf territories was also strongly correlated with increased depredation risk. In the anthropogenic characteristics category, we talked about human land use and management actions that influence wolf behavior and livestock vulnerability. Distance from human residents increased depredation risk. Farms with lower road density and fewer croplands were associated with chronic depredations. Lethal removal did not reduce long-term depredations, so targeted removal of specific pack members may reduce future depredations. Non-lethal measures show variable success. For example, flaggery, which is a visual barrier of brightly colored flags or ribbons attached to a wire, was shown to exclude wolves for up to 60 days with extensive maintenance. Increased human presence and activity also similarly decrease depredation risk, although this effect may be context dependent. In conclusion, although verified depredation events remain relatively rare on a per farm basis, the number of affected farms is increasing with wolf range expansion and changes in land use. Certain covariates, such as larger pasture and herd size, distance from human residences, and proximity to wolf dens, were consistently associated with increased depredation risk. Other factors, like carcass disposal methods and wolf mortality, produce more equivocal results. In terms of management, there are several steps that livestock producers can take to limit livestock losses due to depredation. Prioritize monitoring in high-risk areas. Risk mapping and surveillance during peak depredation season can help producers predict conflict. Areas with large herds, low human presence, and historical depredation events should be monitored closely. 2. Improve animal husbandry practices. Practices such as shifting grazing closer to human dwellings, increasing human presence in vulnerable pastures, and disposing of carcasses properly may help decrease depredations. 3. Use non-lethal deterrents. Flattery, electric fencing, and guard dogs can offer temporary protection, especially in smaller pastures. However, these methods require proper deployment and regular maintenance to be as effective as possible. Use lethal control Number 4, use lethal control cautiously and strategically. Targeted removal of problematic individuals may decrease recurring depredations. And 5, adapt solutions based on local conditions. Factors such as local prey abundance, landscape structure, and pack behavior must be considered in any depredation prevention strategy. As wolf livestock conflicts increase across the West, more research is needed to understand the temporal and spatial variability of these conflicts. Many Western states, such as Oregon, collect data on depredations by wolves, but have not examined the factors that contribute to these depredations. Our literature review identified four broad categories of covariates: wolf pack characteristics, climate and environmental characteristics, livestock operation characteristics, and anthropogenic characteristics that have been associated with depredation risk. Results from this literature review will be used in the future analyses examining wolf depredations of livestock in Oregon. I would like to acknowledge my mentor Lara Menjak and Oregon State University for facilitating and assisting with this literature review. I would also like to acknowledge the Harvard Open Bio undergraduate lab for providing me with the opportunity to conduct this review. Thank you.
Awesome. I really enjoyed listening to that, and I enjoyed the topic. Definitely some potential here, for solutions in the future. All right. This will be our last presenter before the break. Um, let's hear it from Vagmi for biomarkers in neurodegeneration. So actually somewhat similar to what I did last year.
Good afternoon, everyone. I worked with my mentor Wei Liu of UC Berkeley on this literature review, and today I'll be talking about my research on blood-based biomarkers and exosome-driven biomarkers for Alzheimer's and Parkinson's disease. Both Alzheimer's and Parkinson's are progressive neurodegenerative disorders that cause damage to the brain over many years. Alzheimer's is best known for memory loss and cognitive decline, while Parkinson's mainly affects movement, causing tremors, stiffness, and slowed motion. Both diseases involve chronic inflammation in the brain, buildup of abnormal proteins, and the gradual loss of healthy neurons. Currently, diagnosing these conditions with high certainty often requires invasive or expensive tests, like lumbar punctures to analyze cerebrospinal fluid or PET scans to visualize protein buildup in the brain. These tests work, but they're not practical for routine screening or for monitoring patients frequently. This is where blood-based biomarkers come in. Blood biomarkers are measurable molecules in the bloodstream that give us clues about what's happening in the brain. Some circulate freely in the plasma or serum, while others are carried in exosomes, which are tiny vesicles released by cells. You can imagine exosomes as mini delivery trucks that transport cargo such as proteins, lipids, and RNA from one cell to another. When brain cells release these exosomes, they carry molecular snapshots of what's really going on inside these cells, and by capturing them from blood, we can get a window into the brain without directly sampling it invasively. For our research, we conducted a targeted narrative review. This means we didn't try to collect every paper ever written. Instead, we started with a set of recent, highly cited review articles, identified the most relevant and well-designed studies they referenced, and then traced those primary sources back to the original data. This approach let us focus on the highest quality evidence, understanding the biological mechanisms, and also recognizing where the gaps and inconsistencies were. I focused on three main categories of biomarkers: microRNAs, lipids, and proteins. MicroRNAs or miRNAs are short RNA molecules that act as regulators of gene expression. They can turn the production of certain proteins up or down. In Alzheimer's, some miRNAs that normally help control the processing of amyloid precursor protein are reduced, leading to increased production of amyloid beta, which is the main component of plaques found in Alzheimer's disease. Others influence tau, the protein that forms tangles in Alzheimer's brains. In Parkinson's, certain mRNAs that are involved in the inflammation or health of dopamine producing neurons, meaning changes in their levels can reflect disease processes. Lipids are another important category. They're key components of cell membranes and also act as signaling molecules. In Alzheimer's, changes in specific lipids like ceramides may indicate that cells are under stress or undergoing apoptosis, the programmed process of cell death. In Parkinson's, lipid changes can reflect problems in ways that cells recycle damaged
components, a process known as autophagy, which is disrupted in the disease. Proteins are currently the most advanced biomarkers for clinical use. In Alzheimer's, form of tau proteins that have been chemically modified, such as phosphorylated tau, are highly specific indicators of the disease. Elevated levels of p-tau 217 in the blood are strongly associated with Alzheimer's pathology. In Parkinson's, the main protein of interest is alpha-synuclein, which clumps together to form Lewy bodies in the brain. Measuring alpha-synuclein in neuron-derived exosomes rather than in whole blood improves the ability to distinguish Parkinson's patients from healthy controls because it focuses on brain-derived material instead of signals from the rest of the body. Across all of these, exosomal biomarkers often provide a clearer and more specific signal than free-floating molecules in the plasma or serum, but they require more complex laboratory techniques to isolate and measure. The potential here is enormous, because imagine being able to detect Alzheimer's or Parkinson's years before the symptoms begin using just a blood test. This could make it possible to start interventions earlier for millions of people, track disease progression more accurately, and select the right participants for future clinical trials. But my review also made it clear that we're not there yet. Different studies use different methods to collect samples, isolating exosomes, and measure biomarkers, making it hard to compare results. Many studies also have smaller sample sizes, and findings in one population don't always hold true in another. So the next steps are clear. We need standardized protocols for biomarker measurement, larger and more diverse participant groups, and longitudinal studies that follow people over time to see how these biomarkers change as diseases progress. In short, my research shows that blood-based and exosomal biomarkers are a promising bridge between complex brain processes and accessible testing methods. They have the potential to transform diagnosis and monitoring, but be careful because coordinated research is essential to make that vision a reality. Thank you to Harvard Open Bio SRI for this amazing opportunity, and I'm so grateful for the support I've received from my mentor as I embarked on the literature review process.
Awesome. I think your explanations were very well put. And I actually understood a good amount of that. Okay. So this was the last presenter we had scheduled before the break, but I'm going to attempt to try to get this video to work. So please bear with me if something goes wrong. Message in the chat. But I believe I have to unplug my headset for the video to play. If it doesn't work, feel free to send something in the chat and I will see it still. All right, we're going to try this.
Hi everyone. So my research is about evaluating waveform parameters for efficiency and efficacy in micromagnetic simulation. So in magnetic simulation, or micro-MS for short, um, it's a promising new technology in the field of neuromodulation, and unlike traditional electrical simulation, which requires direct electrical contact with tissue, micro-MS uses magnetic fields to induce electrical currents, which avoids that galvanic contact. So this avoids issues like biofouling, which previously occurred in electrical stimulation, and then it also allows for the same really precise therapy on specific neural circuits. ICROMS, however, it faces like a major technical challenge, which is to effectively simulate neurons. So the implanted microcoils require high electrical currents, and this takes a lot of power, which limits battery life for any devices that are like implanted in the brain, and that can end up like burning up the implanted coil because of how much heat is generated. And that heat is obviously a major safety concern too, because it can potentially damage neural tissue. So my literature review is trying to understand how we can make micro-MS more effective and more efficient. So first, we can look at my methods section, which shows how I first got my eligibility criteria. So I looked for only peer-reviewed original works, which are English. And then I focused on articles about like electrical current waveforms and power efficiency and heat generation because like a lot of the works didn't talk about the waveforms enough. And then I searched for terms And then I ended up getting 68 records, which I used in my literature review. And then you can look at the findings section next. Um, so this is our findings on this like literature, and it shows like how micro MS is a much less mature like technology compared to transcranial magnetic stimulation or TMS. So we decided to look into like literature which focuses on the waveforms in TMS so we can adapt it to the waveforms in micro MS. So you can see on on table one, we looked at first the waveforms already in micro-MS. So we looked at sinusoidal waveforms, triangular waveforms, square waveforms, and sawtooth waveforms. And these like are electrical currents that we pass through this microcoil inside the brain that's implanted in the brain. And then this current that goes through the coil will generate this magnetic field that like goes in this like really focal point in your brain uh, and it passes through like nerves, right? And then those nerves then get an electric field, and their neurons trigger action potentials, which basically stimulates the brain, right? And so we pass these four different types of electrical currents through that microcoil inside the brain. And those are the currently used waveforms in micro MS, in the field of micro MS. And then you can look at table two, which is the review I did over TMS waveforms, and it shows the monophasic, biphasic, trapezoidal, and then near rectangular electrical field inducing waveform. So the first three, those are electrical current waveforms as well, which pass through a coil in TMS. And TMS is a little different from micro MS because it's at a much larger scale and there's no microcoil implanted in the brain. So it's completely non-invasive. And in TMS, all it does is it has an external coil where this waveform is passed through, and that external coil creates a larger magnetic field, which then triggers action potentials in neurons. And TMS is much less precise than micro MS, which is why we're looking at waveforms in TMS that we can adapt to the less mature field of micro MS. So the fourth one, which is called a near rectangular E field waveform, um, it is a waveform that's either monophasic or triangular, and this waveform um, then like is optimized. It's like it's optimized through like software so that the resulting magnetic field or sorry electric field that finally triggers action potentials is a near rectangular shape. And this waveform was had a study on it that showed that it gave it used much less energy and generated less heat from the coil. And this is something that future research can use in micro MS. Like it can be adapted to micro MS to be used in that field as well. So um, in conclusion, this review underscores that waveform parameters, like they profoundly impact the efficiency and efficacy of micro-MS, which is shown through the original sinusoidal, triangular, square, and sawtooth waveforms. It shows how different heat generated, different power generated, and different like effectiveness is shown with different like parameters, such as like pulse width or frequency or amplitude and other parameters as well. And then it also shows how my literature review also talks about how micro MS is MRI compatible and it has high precision, but it also has a lot of risk with it, with burning up the coil, right? So we talked about any any TMS waveforms that could be used um, in micro MS, and in future works these will be used, right? And then finally we talked about how um, that like that impacts micro MS, how This is going to support anybody else doing research in microMS and trying to lower the heat generated and the power of and make the power usage more efficient. Um, so yeah, that's my research. I also wanted to thank you guys for giving me this opportunity to do this research and to present to you guys. So thank you.
I'm trying to get out of full screen. There we go. All right. All right. So that being put, we're gonna start our break now. Feel free to get up, go to the bathroom, go grab a snack, do whatever you want, but make sure to be back here. We're gonna start with the next presenter at 1:36 p.m. Eastern Time sharp. So yeah, try to be back here a couple minutes before that. But yeah, we will resume then. All right, if everybody's back, I'm gonna ask that you could all please turn on your cameras, so I know who's all back. Um, and yeah, we're gonna start this next presentation in a couple of seconds whenever it hits 1:36. All right, the clock has hit 1:36. We're going to go on with Shaunak talking about the dark proteome. Excited to learn about it.
Awesome. Good afternoon, everyone. My name is Shaunak, a rising senior at Menlo Atherton High School in California, and I'm super excited to share my project, which is decoding the dark proteome, deep learning-enabled discovery of druggable enzymes in Wucheria bancrofti. During the summer, I went from tens of thousands of unknown proteins in this neglected tropical parasite to precise functional annotations and computationally designed inhibitors. And while there's a lot more to do, this could be the first step towards an entirely new treatment for a disease that affects hundreds of millions of people globally. But starting with some background, W. bancrofti is a parasitic roundworm, as visualized in the top left here, that causes lymphatic filariasis. You might know it by its most severe form, elephantiasis, where limbs swell and disfigure permanently. Over 36 million people are already permanently disabled, and more than 657 million more are at risk across 39 countries. The biggest problem is that current drugs cannot kill reliably adult worms. These worms live for years in the lymphatic system, causing damage and spreading to others. The World Health Organization has explicitly called for new drug targets to change this. But there's a major roadblock. Over 90% of the worm's proteome is quote unquote dark, meaning we have the amino acid sequences, but we have no idea what they actually do. Without knowing the function, we cannot design drugs against them. Enzyme commission, or EC numbers, are a way to assign function to enzymes by the very specific reaction they catalyze. The only way that enzymes get cataloged in this database is if they are experimentally tested, which results in a large gap in annotation as these things take time. My work was to take this dark proteome and do three things. First, precisely predict the enzymatic function of these unknown proteins. Two, identify the most druggable ones. And three, find small molecule inhibitors to target multiple pathways on this parasite. The novelty here is that we engineered specialized state-of-the-art deep learning models which enable us to discover hundreds of enzymes and multiple drugs, which has never been done before in this parasite. As visualized in the top right in our methodology and deep learning architectures, we developed the following novel workflow. First, we performed enzyme binary classification. In the W. bancrofti proteome of 48,000 proteins, we know around 4,000 of the functions, which means the remaining 90%, or around 44,000, have unknown functions. We adapted and designed a novel protein detection vision transformer to classify whether each sequence is likely to be an enzyme. We produced one confidence probability per sequence by using a compact evidence aggregation head that converts multiple proposals into a single probability of enzymatic function denoted as probability of enzyme, or P enzyme. Only predictions with greater than 98% probability or 98% confidence advanced to the next stage. Step two was precise function assignment. The ones that had more than 98% were passed down to a hierarchical nearest neighbor model for easy number prediction. We fine-tuned hi-fi NN on parasite proteins, then applied rejection sampling to keep only predictions with 100% model confidence at the full four-level EC number. Step three was target prioritization. After identifying novel classes and a bunch never reported for in W. bancrofti, we prioritized targets using four criteria. First is parasite specificity, how similar it is to the human host. If it was more than 30% similar, it would be disregarded, as that means there's a high chance and high risk of off-target toxicity, and we accidentally target our own human enzymes. Second is chemical tractability. The enzyme had to have known small molecule inhibitors against it. Third, it had to be important in the parasite. It had to be a relevant protein, as disrupting it would have to significantly affect the parasite and its functions. And four, it had to be biologically plausible. We could verify this enzyme probably actually existed in the parasite through its existence in nearby neighbors, such as Brugia or other roundworms, so it wasn't a complete hallucination of the model. And the final step was inhibitor selection and binding prediction. For our top targets, I curated a compound inhibitor library from Campbell and BindingDB, and using both two cofolding, I predicted how each inhibitor and drug would bind to the proteins, which means that we would be able to see the potency and the affinity of each inhibitor against the proteins. Our results are as follows in the bottom left. In total, our fine-tuned model outperformed any other model for W-Bancrofti and is the state-of-the-art model. Also, 14,772 proteins were annotated from the unknown, and 543 new EC classes were discovered for W-Bancrofti. And also, 6 top drug targets were selected spanning 5 strategies. Drug potency was tested against each EC class in the line graph shown below, where a lower graph is a stronger affinity. All of its targets had a predicted affinity under 1 micromolar, which is the standard for identifying a hit candidate. And what's exciting here is that we annotated proteins, but also produced ready-to-test drug candidates. This is the first time that the W-Bancrofti dark proteome has been functionally mapped at this scale with such high precision, and the first time these functions have been directly tied to druggability within the same computational workflow. The next steps are expressing these targets in the lab and testing it experimentally, and using AI-driven generative design to refine compounds for parasite specificity. Thank you to my mentor, for Jefferson Hernandez, for guiding me through this project, and to all of you for listening.
Wow, that was exceptionally strong. And it appears to be an extremely impactful find and research, and I look forward to where this leads us. All right, so next we're going to have Jeepreet talking about it'll appear cannabis.
Good afternoon, I'm Jeev, a rising senior from Dublin High. Starting on the background in the top left, cannabidiol, or CBD, is a compound found in the cannabis plant that doesn't cause the intoxicating high produced by THC, unlike cannabis. Among older adults, CBD use is growing, often to help with pain, sleep issues, or anxieties. But studies have found that CBD use often overlaps with cannabis use, which means THC exposure, that can affect memory, coordination, and reaction time, increasing the risk of drug interactions, especially for those on multiple prescriptions. In 2022, about 14% of older adults reported using CBD, yet national studies haven't separated CBD-only users from those who also use THC-containing cannabis. Our study aims to be the first to make that distinction. On to research questions in the left center. So we have 2 research questions guiding our analysis. 1, what's the prevalence of CBD plus cannabis co-use among US older adults with the latest NSDUH data? And 2, what characteristics distinguish CBD only from CBD plus cannabis users? Here's how we answered those questions. Methods on the lower left. We use data from the NSDUH, the National Survey on Drug Use and Health, a large nationally representative survey of the US population. Our sample consisted of responses from 2022 and 2023 adults aged 65 and older who reported using CBD in the past year. That gave us just over 1,500 people in total. On to measures. For research question 1, we used 2023 NSDUH data to classify older CBD users as CBD only or CBD plus cannabis excluding CBD or hemp. For research question 2, we pooled both years, comparing demographics, physical function, mental health, and other substance use, then modeled these factors to identify characteristics linked to co-use. Analyses were all conducted in R. We calculated nationally weighted descriptive statistics for our major outcomes using the survey and Surveyor packages and compared groups with Rouse-Scott adjusted chi-square tests. We then fit a survey-weighted logistic regression in the VGEM package, which applies NSDUH's complex sampling weights so results generalized to the entire US adult population. The outputs from these models produce the figures you see in this poster. Now let's move on to the first graph in the bottom left, answering our first research question. This graph is a bar chart showing the percentage of older adults who use CBD only versus CBD plus cannabis in the year 2023. This shows that about 9% of older adults use CBD only and just under 6% use CBD plus cannabis. That's over 2 million people nationwide. This is the first national estimate for co-use in this age group and shows that demographics of older adults who co-use is not insignificant. Moving on to the bivariate comparison in the top center. This is another bar chart, but here each bar represents a characteristic, for example, being female, tobacco use, alcohol use, memory difficulty, and how common that characteristic is in CBD-only users versus co-users. From this, CBD-only users are much more likely to be women. Co-users have higher rates of tobacco use, alcohol use, and prescription opioid misuse. There's a small difference in memory difficulty, slightly higher in the CBD-only group. On to the forest plot in the bottom center, answering our second research question. Here's how to interpret the forest graph. Each dot is an odds ratio. Each horizontal line is a 95% confidence interval, and the red dashed line, vertical line at 1 means no difference. If the whole confidence interval is to the right of the line, the factor is linked to CBD only use. If it's to the left, it's linked to co-use. Our major results are that women are much more likely to be CBD only users, and that tobacco use, alcohol use, and prescription opioid misuse are all linked to higher odds of co-use. So moving on to the discussions in the top right. So what does this tell us? Co-use isn't rare. Nearly 6% of older adults, or over 2 million people, use both CBD and cannabis. Men and those with other substance use are much more likely to co-use. We didn't find strong differences in physical function or depression, and once other factors were taken into account. So moving on to the strengths and limitations. Strengths here are that this is recent, nationally representative data, and it's the first US study to separate CBD only from CBD plus cannabis use among older adults. The limitations. One, it's cross-sectional, not longitudinal, so there's no cause and effect, or at least proven. There's no information on pain or sleep, which we know are important reasons for co-use. There's no THC or CBD potency data, and there's no information about cannabis-only users in this data set. All are necessary for future directions. And moving on to future directions, so where do we go from here? First, expand beyond CBD users to compare CBD only, CBD plus cannabis, and cannabis only groups. Second, add more detailed measures like pain, sleep quality, product content, dose, and route of use so we can better understand the motivation and effects. Third, build a causal diagram, a map showing how all our predictors connect to CBD or cannabis choice. That would help future studies design better recruitment strategies and account for confounders. So in short, in 2023, over 2 million US adults who use CBD also use cannabis. Understanding who these people are and how they differ from CBD only and cannabis only users is the foundation for designing safer studies and giving better clinical guidance. I'd like to thank my mentor, Dr. Lamatanya, for her invaluable help, and Dr. Fillingham for his advice. My mentor was supported by the NIH grant. Thank you.
Awesome. I think that's great, and I really do see how this could be the foundation of nationally representative work to come. All right, next we have Caroline talking about disparities in prognostic tools for, once again, breast cancer.
Okay, hi everyone. My name is Caroline, and I'm a rising senior at Limbrook. And my mentor is Hannah Cessna, who's a PhD student at Stanford. And today I'm excited to share the research we did on disparities in prognostic tools for breast cancer, specifically how polygenic and recurrence risk scores can underperform in certain populations due to differences in race, ethnicity, and tumor biology. So to start off, breast cancer is one of the most common cancers worldwide, with over 2 million new diagnoses each year. And survival rates have improved thanks to advances in early detection and treatment, but they have not improved equally for everyone. Women of color, including Black, Hispanic, and South Asian women, often face worse outcomes due to a combination of biological, social, and structural factors. One tool that doctors use to guide treatment is a prognostic model, which includes tests like Oncotype DX or polygenic risk scores, also called PRS. These tools predict how aggressive someone's cancer is or how likely it is to come back. And based on the results, doctors decide whether a patient should get chemotherapy, hormone therapy, or just be monitored. The issue is that many of these tools were developed and validated using mostly European data, which means that they don't always work as well for patients from other racial or ethnic backgrounds. In other words, the very tools designed to personalize cancer care might be reinforcing existing disparities. To understand why, let's take a look at figure 1, which you'll find at the bottom of my introduction. This diagram shows the main subtypes of breast cancer, which are defined by the presence or absence of hormone receptors for hormones like estrogen and progesterone, as well as the protein HER2. Luminal A is the most common subtype, and it has the best prognosis and is more common in white women. On the other hand, triple negative breast cancer, or TNBC, is more aggressive and disproportionately affects Black women. So prognostic tools may not fully account for these subtype differences or how they interact with race. For example, a model trained mostly on luminal A tumors in white populations might struggle to predict outcomes for TNBC in Black women. To explore these disparities further, my project synthesized findings from multiple studies on how well current prognostic tools perform across racial and subtype specific groups. And I focused on 3 key themes, which were PRS performance, recurrence risk score disparities, and subtype specific prognosis. So first off, I found that PRS often underperform in non-European populations because of differences in genetic markers like allele frequencies or linkage disequilibrium patterns. And even with multi-ethnic reference panels, PRS tend to be less accurate for African and Asian ancestry groups. And these inaccuracies can have real-world consequences since overestimating risk can lead to unnecessary treatment, while underestimating risk can lead to delayed preventative care. Similarly, recurrence risk scores like Oncotype DX also show racial disparities. One study found that they were strong predictors for white women, but less accurate for Black and Hispanic women. In addition, patients in rural or low resource hospitals may never even be able to receive this genomic testing in the first place, especially if their providers aren't properly trained to use them or if their insurance doesn't cover it. And even when these tests are available, these models can be hard to interpret for underrepresented groups if they weren't validated in more diverse populations to begin with. Finally, certain subtypes are more aggressive and more common in some racial groups, yet underrepresented in model training data sets. This reduces generalizability and risks misclassifying patients, even among those with the same subtype. In fact, some studies found ethnic disparities in survival outcomes within the exact same breast cancer subtype, which suggests that social and structural factors are major contributors. Overall, prognostic tools are really powerful and can be extremely useful, but only if they're accurate and equitably applied. If not, they can contribute to harms like unnecessary treatment or missed opportunities. And to improve this issue, we need more diverse training data, better validation across ancestry groups, and models that integrate both biological and social determinants of health. And by addressing these gaps and fostering more collaboration between researchers, clinicians, and policymakers, we can ensure that these prognostic tools work for all patients and advance toward a more personalized breast cancer care. And I would like to thank my mentor for her guidance and Harvard SI for the opportunity to do this research. Thank you.
Awesome. Yeah. I think that was good research and definitely a good stepping stone for many, many papers to come in the future. All right, next up we have Wesley, and they'll be talking about dopaminergic mechanisms relating to schizophrenia.
Good afternoon, everyone. As part of my research, I conducted a narrative literature review examining current evidence on how the D2 and D3 dopamine receptors are involved in schizophrenia, synthesizing findings from genetic, expression, and imaging studies alongside their therapeutic relevance. Schizophrenia is a profoundly complex psychiatric disorder, affecting roughly 0.5% of the global population. It is defined not by a single set of symptoms, but by a constellation of positive, negative, and cognitive impairments. Positive symptoms, hallucinations, delusions, disorganized thinking, can be disruptive in obvious ways. Negative symptoms like avolition, alogia, and blunted emotional expression are more insidious, often resistant to treatment, and can erode a patient's ability to function long after acute psychosis subsides. One of the most enduring frameworks for understanding this disorder is the dopamine hypothesis. In its earliest form, it proposed that excessive dopamine activity caused psychosis, without distinguishing between symptom types. Later refinements introduced a regional model, excess dopamine in the striatum driving positive symptoms and reduced dopamine in the frontal cortex contributing to negative symptoms. Today's version 3 shifts the focus away from receptor overactivation, but more towards presynaptic dysregulation, basically abnormal synthesis and release of dopamine. Within this context, two receptors have commanded particular attention, D2 and D3, encoded by the DRD2 and DRD3 genes, respectively. Both are members of the D2-like family of the G protein-coupled receptors, and they share structural similarity but diverge sharply in distribution and function. D2 receptors are abundant in the dorsal striatum and medial temporal regions, while D3 receptors concentrate in the ventral striatum, an area for reward, motivation, and emotion. D3 receptors also have a higher affinity for dopamine and may be more sensitive to disruptions in glutamatergic signaling. The question is, how do these receptors behave in schizophrenia? Here, the literature becomes messy. Early theories treated D2 upregulation as a biomarker, yet modern PET and postmortem studies disagree. Some show increases, others reductions, and still others no change. Certain DRD2 polymorphisms like serine 311 cysteine or -141C in STEL have been linked to schizophrenia in some populations, but often fail to replicate. Even receptor conformation can shift. with atypical D2-like binding sites appearing in patients but not controls. The D3 story is equally tangled. The serotype glide polymorphism, which alters binding affinity, has been associated with risk in some European cohorts, but not in Han Chinese or other populations. Expression data are contradictory. Some studies report elevated DRD3 mRNA in blood, others loss in specific cortical areas, and others no change. Abnormal splicing events producing non-functional D3 isoforms have been found in the anterior cingulate cortex. DRD3 expression has even been linked to symptom patterns, higher levels with negative symptoms, lower with positive symptoms. Therapeutically, these receptors remain crucial. Clozapine, the gold standard for treatment-resistant schizophrenia, binds D2 and D3 in a distinct cortical to striatal pattern that may spare motor side effects. Amisulpride's dose-dependent selectivity can target either negative or positive symptoms. Brexpiprazole, a partial D2-D3 agonist, offers balanced modulation for long-time stability. Beyond dopamine, D2 adenosine A2A interactions and D3 modulation via cannabidiol suggest novel interventions. If there's one unifying theme, it is inconsistency. The picture of D2 and D3 in schizophrenia is fragmented and often contradictory. That is not a reason to abandon the investigation, it is a reason to sharpen it. Future research must integrate multimodal approaches, pairing genetic data with expression profiles, mapping receptor occupancy with symptom trajectories, and examining gene-gene interactions like DRD3 with BDNF. Studies must also account for ethnic variability and haplotype context, rather than treating single polymorphisms in isolation. Only through layered, population-aware, mechanistic research will we begin to reconcile these contradictions and move closer to treatments that address not just the symptoms, but the underlying neurobiology of schizophrenia. I would like to thank my mentor and Open Bio Lab for this opportunity. Thank you.
Oh, awesome. I think that was very well put. And I like that you identified the kind of disparities of how they treat those two receptors. All right. Next we have Srishanth. Hopefully I'm saying that somewhat correctly. Talking about chronic cocaine use.
Yeah, so I'm Srishant. My mentor is Tate Erickson, and I will be presenting about longitudinal myelin mapping of the macaque frontal lobe across chronic cocaine use and abstinence. So chronic cocaine use is a very significant public health crisis because of its lasting impacts on brain structure, changing synaptic plasticity and impairing the prefrontal cortex. So past research has shown that white matter integrity decreases as a result of cocaine exposure, and myelin is a key component of white matter. So myelin is really important because it allows for rapid signal transmission and cognitive function. So disruption to it can cause dysfunction and other impairments to the brain. Current limitations include lack of longitudinal studies tracking myelin changes specifically, and then also a heavy reliance on rodent models, which are different compared to humans other than non-human primates or NHPs. And there's been very limited access to ultra-high field MRI. So our research question was, how does cocaine exposure affect myelin content and recovery in the frontal lobe over time? So to do this, macaques were placed in an operant conditioning environment with cocaine self-administration protocol, and 10.5 Tesla MRI was used to get the T1 weighted and T2 weighted MRI scans at 3 time points. T0, which was the before cocaine exposure, T3, which was during cocaine exposure, and T6, which was during abstinence. Following this, we used FSL and AFNI to pre-process our images, and then we generated our myelin maps. And then after that, we calculated summary statistics. As you can see in table 1 in the top right corner of the results section, we calculated mean, standard deviation, standard error of the mean for all 3 time points. And then figure 1 shows the projections of the mean myelin content. And we did this by extracting the mean from the table and then projecting using SUMA. In addition to the summary statistics, we also calculated change and percent change in T3 and T6 from T0, and you can see this in table 2. Figure 2 shows the projections of the change in mean myelin content and the percent change in mean myelin content, and we projected this in the same way as figure 1. So using our surface projections and our data tables, we found 4 main distinct patterns of change. The first one was recovery, which occurred in areas 32, 24 in the MCC, where early myelin loss happened during exposure, but then restoration of that myelin happened during abstinence, which shows that recovery potential is possible. Sustained increase was the next pattern, where regions such as the frontal pole showed progressive myelin increase the entire time during exposure and abstinence. And the next pattern was continuous decline, which was the exact opposite of sustained increase, where regions such as the M1 and SMA pre-SMA regions showed chronic disruption of myelin with progressive demyelination during exposure and abstinence. And lastly, their last pattern was biphasic response. Interestingly, myelin actually increased during cocaine exposure and then decreased during abstinence. And this could happen because of plasticity in the form of compensation, where early glial activation occurs to counteract the cocaine use, but then net demyelination occurs. So our findings align with prior work of myelin plasticity and vulnerability, and they show that chronic cocaine use leads to non-uniform, time-specific, and region-specific myelin changes in the macaque frontal lobe, and that neural pathways and circuits each respond uniquely. So this research contributes to the first longitudinal high-resolution mapping of frontal myelin content in a macaque model, and future work could extend this analysis to whole brain myelin mapping and also across all imaging time points. And also, we chose to perform a group level analysis, but future work could focus on subject level analysis to see the differences hidden by the group. We're also very interested in analyzing intrinsic neural timescales, or INTs, to see how temporal processing and structure function coupling is affected by cocaine use. And the areas that showed the recovery, which were area 32, 24, and the MCC, there can also be more explorations to see the molecular basis for why that pattern actually happened. And lastly, behavioral tasks can be used to correlate to myelin changes to see how decision-making, risk of relapse, impulsivity, and other impairments are affected by cocaine use. So I'd like to thank Tate Erickson for his invaluable mentorship throughout this entire project, and also the SRI team for giving me the opportunity to conduct this research and present it here. Thank you so much.
Yeah, I think that was really well put. I followed you through most of that. Um, yeah, really impactful work, and I'm very excited to see where this leads, as you mentioned, aligning with previous research. All right, next up we have Rita talking about metabolic maturation of cardiomyocytes.
Hi, my name is Rida Hajwane, and I'm a rising senior at Parsippany High School located in Parsippany, New Jersey. My mentor, Frank Ketchum, and I have constructed a systematic literature review on the metabolic maturation of human-induced pluripotent stem cell-derived cardiomyocytes, which are also known as HIPSCCMs. This literature review describes the great advances and ongoing challenges in inducing maturation of HIPSCCMs to more closely resemble adult cardiomyocytes. While HIPSCCMs have great potential for disease modeling, pharmacological testing, and regenerative medicine, their immaturity limits their application at this time. Morphological enhancements including increased cell size, elongation, improved myofibrillar organization are always observed with long-term culture. Physiologically matured HIPSCCMs exhibit enhanced contractile function and improved calcium handling. Additional innovation in these areas is significant to overcome present limitations and achieve full therapeutic potential of HIPSCCMs in the cardiovascular field. To start off, HIPSCCMs are known as an innovative resource for cardiovascular research, offering a system to address cardiac development, model inherited and acquired heart disease, and facilitate drug discovery and toxicity screening. The ability to generate an apparently unlimited supply of patient-specific cardiomyocytes from HIPSCs is of vast potential in revolutionizing personalized medicine and developing novel cell-based therapies. However, one persistent key hindrance to the full realization of this potential is the inherent immaturity of HIPSCCMs. These cells often recapitulate characteristics more closely similar to fetal or neonatal cardiomyocytes than to their mature adult counterparts. Such immaturity is multifaceted, manifesting as structural disorganization, suboptimal electrophysiological behavior, and a predominantly glycolytic metabolic phenotype, an extreme contrast to the adult cardiomyocytes' robust oxidative metabolism and highly organized structure. This literature review was conducted by searching and synthesizing information from a set of uploaded documents systematically. The strategy for the search was to search for articles on the maturation of HIPSCCMs with an emphasis on culture media and metabolic modulation strategies. We learned cautiously about reporting information impacts of such metabolic therapies on cell phenotypes, like enhanced contractile force, sarcomere length, regulation of calcium, and adult cardiac gene expression. The review also covered experiments that took these metabolic maturation strategies to more mature experimental models like engineered heart tissues to assess their effectiveness in more physiologically relevant conditions by integrating data from all the relevant sources. This review aims at presenting an intensive but summary overview of the state of the art. so far emphasizing both the gains as well as the current limitations towards functional maturity attainable at HIPSC-CMs for translation purposes. Below is also a picture of a male HIPSC-derived cardiomyocyte. Experiments using special maturation media have demonstrated phenomenal improvement in the structural properties and physiological function of HIPSC-CMs compared to cells cultured under standard conditions. Specifically, prolonged in vitro culture, usually more than 80 to 120 days, was shown to induce phenomenal morphological changes in HIPSC-CMs. These include larger cell size and length, higher percentage of multinucleated cells, and more developed myofibrils with visible sacromeres. Ultrastructural investigations have supported improvements in myofibril density and alignment, with late-stage cells possessing more organized sarcomeres and larger lengths toward adult values. Functionally, these older HIPSC-CMs also exhibit more contractile function with a twofold increase in shortening amplitude and improved calcium handling with faster rates of calcium release and reuptake. Electrophysiological characterization has demonstrated more adult-like properties with hyperpolarized maximum diastolic potentials and faster action potential upstroke velocities, along with greater expression of key genes like KCNJ2 and CSN5A. To conclude, this literature review highlights key strides made in maturing HIPSC-CMs towards an adult-like state, indispensable for their viability in disease modeling and regenerative medicine. Despite huge strides, current maturation protocols, while improving metabolic properties, structural organization, and electrophysiological activity, remain far from mimicking adult cardiac tissue complexity. The switch towards fatty acid oxidation as the primary energy source and the enhancement of mitochondrial function are key determinants of successful metabolic maturity, but to be completely metabolically flexible is an ask too big. And lastly, I would like to thank Open Bio Laboratories and the Student Research Institute for this opportunity, and my mentor, Mr. Frank Ketchum, for guiding me and helping me throughout this program. Thank you.
Awesome. I commend the nuance that went into this and the coverage of the pluripotent stem cells. Next up, we have Naitik talking about, once again, non-human primate subjects and also cocaine.
All right, good afternoon, everybody. My name's Naitik Mohanty, and I go to Sandra Day O'Connor High School located in Phoenix, Arizona, and I'll be talking to you about cocaine addiction. So, cocaine use disorder produces progressive changes in brain networks involved in reward, executive control, and habit formation. The putamen part of the dorsal striatum integrates dopaminergic input with motor and cognitive signals, playing a central role in habitual drug seeking. Most studies provide single time point input data, often analyzing cross-sectional studies, leaving the temporal trajectory of these changes unclear. So to fill this gap, we conducted a longitudinal resting state fMRI study in non-human primates to track how putamen-centered connectivity evolves over early, mid, and late stage cocaine self-administration and addiction. So how we did this includes 4 macaque monkeys self-administered intravenous cocaine under a intermittent access schedule around 5 days per week. Scans were required at a baseline and after 5 days after, 45 days after, and 95 days after of drug exposure using a 10.5 Tesla MRI for high resolution structural and functional data. So we used seed-based connectivity analysis in the CONN toolbox, that's the CONN toolbox, with the putamen as the seed region. Data pre-processing included denoising, motion correction, distortion correction, and spatial normalization to the NIMH macaque template. Additionally, group level analyses applied threshold-free cluster enhancement with permutation testing as well as FDR correction. So a result, day 5 saw significant increases in connectivity between the putamen and the medial prefrontal cortex, the anterior cingulate cortex, the dorsolateral prefrontal cortex, as well as various other supplementary motor areas. This reflects early recruitment and essentially taking over of reward and control pathways. Day 45 expanded these increases to the orbital frontal cortex, the ventrolateral prefrontal cortex, the insula, and other motor areas as well. However, we saw the first decreases appear in the parietal cortex and the posterior cingulate cortex, indicating early disruption in the sensory integration and default mode networks. Day 95, interestingly, saw the connectivity reductions dominated in the precuneus, the posterior cingulate cortex, the temporal cortex, and the visual cortex, essentially showing that frontal hyperconnectivity that we saw in day 5 was significantly diminished. So how do we interpret all this? Our findings mainly reveal a two-phase trajectory, the early adaptation phase and the late breakdown phase. The early adaptation phase sees hyperconnectivity in the frontal putamen circuits, and it supports goal-directed drug seeking, essentially, while the late breakdown phase sees widespread posterior and default mode network disruption, aligning with impaired self-monitoring and compulsive use. So why do we care? Well, understanding when and where these network changes occur can inform biomarker development and stage-specific interventions. For example, bolstering executive control networks in early stages and targeting default mode recovery in latter stages. This has the direct implications for treatment timing and relapse prevention strategies in real human addiction. So, in conclusion, chronic cocaine exposure reorganizes brain networks in a predictable sequence. The initial frontal hyperconnectivity followed by the posterior network collapse. Identifying these neural signatures in real time could actually allow clinicians to intervene before irreversible and devastating network degradation occurs. I would like to take some time to thank Harvard Open Bio for the incredible opportunity and letting me conduct this research, as well as my mentor, Dr. Anna Mania, and the University of Minnesota. Thank you.
Awesome. Yeah, I definitely it's kind of cool to see you and I'm sorry, I'm bad at pronouncing these names. Srishant's work kind of coincide with each other. So yeah, that's really neat to see. All right, next up we have Lorelai talking about circadian rhythms with neurodegenerative conditions.
Hi guys, I'm Lorelai Dorsey, and my mentor and me, Sanket Garg, chose to make our review paper on the disruptions in circadian rhythms and how they are central to neurodegenerative conditions. Our research explores how disruptions in circadian rhythms significantly impact several neurological disorders. This includes, but is not limited to, Alzheimer's disease, Down syndrome, Williams syndrome, Smith-Magenis syndrome, and spinal muscular atrophy. Despite differing causes, these disorders share overlapping pathways where circadian dysfunction exacerbates symptoms. During treatment, current treatments range from melatonin and light therapy to gene therapies. We stress that circadian disruption is not merely a side effect but a central factor in disease progression, making the body's eternal clock a promising therapeutic target. The circadian clock regulates our 24-hour sleep-wake cycle, with the master clock located in the brain's suprachiastic nucleus responding to light and temperature cues. This system controls hormones such as melatonin and cortisol, which control sleep and alertness. Sleep supports homeostasis by aiding cellular repair, immune function, brain waste clearance, hormone production, heart health, and stable breathing. When circadian rhythms are disrupted, these critical processes suffer, but this connection often remains overlooked in many conditions. When completing this review paper, I conducted a literature review on circadian disruptions across neurodevelopmental and neurodegenerative disorders. I began by examining circadian system mechanisms, including involved brain regions and genes, then selected a mix of well-studied and less explored conditions. I analyzed research papers to understand each disorder's symptoms, their links to circadian rhythms, and related treatments. Throughout, I synthesized findings and identified gaps to highlight where circadian-focused research should have the greatest impact. We first started in Alzheimer's disease. In Alzheimer's, toxic proteins, amyloid beta and tau tangles, accumulate in the brain. These are normally cleared by the glymphatic system, which operates most effectively during sleep. During this process, the brain's interstitial space expands, allowing cerebrospinal fluid to wash away waste. Sleep disruption slows this clearance, leading to further protein buildup and accelerating disease progression. This creates a vicious cycle where poor sleep worsens Alzheimer's and the disease further impairs sleep. Next, Down syndrome. It results from an extra or partial copy of chromosome 21, affecting development and cognitive function. Many individuals experience obstructive sleep apnea and fragmented sleep, which disrupts essential nighttime restorative processes. This lack of quality rest further impairs cognitive growth and overall development, compounding challenges. Then Williams syndrome is caused by a microdeletion affecting the elastin gene. This leads to elevated nighttime cortisol and reduced melatonin levels. This disturbs deep sleep, and over time, it results in an insufficient restorative of damaged neurons, further compromising intellectual function. Next, Smith-Magenis syndrome arises from the deletion on chromosome 17's P11.2 region, causing RAI1 gene haploinsufficiency. This results in inverted melatonin secretion and significant sleep disturbances, which negatively impact behavior and cognitive abilities. Lastly, with spinal muscular atrophy, it is a neurodegenerative condition marked by muscle weakness due to motor neuron loss. This also disrupts peripheral circadian genes because of an impaired signaling within the body. Consequently, essential bodily processes falter, accelerating muscle deterioration. In conclusion, our review reveals a strong yet unexplored link between circadian rhythm disruptions and neurodevelopmental and neurodegenerative disorders. This is including disorders which were Alzheimer's, Down syndrome, Williams syndrome, Smith-Magenis syndrome, and spinal muscular atrophy. While each disorder has a distinct genetic cause and symptoms, they share circadian disruptions that worsen cognitive decline, behavioral instability, and sleep disturbances, while actively driving disease progression. These disruptions impact vital systems like hormone balance, metabolism, immune response, and waste clearance, which further triggers a cascading complications that disrupt treatment. Despite this, circadian dysfunction remains overlooked in many clinical settings. However, promising treatments such as melatonin supplementation, light therapy, behavioral sleep interventions, and targeted gene or hormone therapies suggest that improving circadian health can enhance the quality of life and may slow down disease progression, even though the cures are not yet available. I would like to specifically acknowledge my mentor, Sankit Garg, for teaching me everything that I pretty much stated while writing this paper and helping me learn how to make review papers to make such a impact on the world based on our studies. And I'd also like to thank Harvard Open Bio for giving me the opportunity to meet my mentor and be able to experience in something like this. Thank you.
Awesome. Yeah. I really did like the kind of like unlinked connection that you talked about before with circadian rhythms to neurodegenerative conditions. It makes sense. And I think most people could understand that. So it's good to simplify that in a sense. Next, we have John talking about the forecasting of waterborne disease outbreaks from atmospheric and environmental indicators. Uh, John, are you here with us? Okay. I do not believe that he is in the room. So let me just check real quick. Go through this. All right. So he is not here. So we will move on to the next presenter. Oh, we have a video. Okay. Let me make sure my sound is shared. All right. Whenever I play this, you guys do a thumbs up, thumbs down, about 5 seconds in to make sure that this is working.
Hello, my name is Tan and today I'll be presenting my project titled Predictors of Radiographic Knee Osteoarthritis Progression in the Osteoarthritis Initiative Cohort. To begin with, knee osteoarthritis, or OA, is a chronic joint disease and one of the leading causes of disability among older adults. While OA can progress slowly over many years, some people experience rapid joint deterioration, which can lead to increased pain, reduced mobility, and higher healthcare costs. Because of this, being able to identify who is at greater risk for faster progression is crucial, as it allows for earlier and more targeted interventions such as weight management, physiotherapy, or even just regular monitoring. With that in mind, the goal of my study was to evaluate which demographic, clinical, and functional factors could predict OA progression over a 10-year period. For this purpose, I used publicly available data from the Osteoarthritis Initiative, a study that followed thousands of participants over 120 months. From this dataset, I included over 3,000 participants who had no or minimal OA at the start, specifically a KL grade below 2 in both knees. Next, I categorized the participants into 3 groups: no progression, slow progression, and fast progression. No progression was defined as a KL grade that stayed below 2 throughout the entire study, whereas slow progression was defined as a KL grade that reached 2 or higher between 84 and 120 months. As for fast progression, it was defined as a KL grade that reached 2 or higher between 12 and 72 months. After establishing these groups, I tested several predictors, which included demographics like sex, age, race, and BMI, patient-reported pain and function using WOMAC scores, and functional performance using the 20-meter walk test measured by both time and the number of steps. To analyze these factors, I applied multinomial logistic regression to determine how each variable was associated with slow or fast progression compared to no progression. Moving on to the results, the big takeaway is that sex was the strongest predictor. Women had nearly double the odds of slow progression and 1.7 times the odds of fast progression compared to men. This is consistent with other research showing that biological and anatomical differences, such as hormonal changes after menopause and differences in knee structure, may make women more vulnerable to OA progression. Second, BMI was the next most significant factor. Each unit increase in BMI raised the odds of progression by 7 to 9%. This finding demonstrates that both the mechanical strength on the joint and the inflammation linked to excess weight can accelerate deterioration. Third, WOMAC pain scores were statistically significant, but their overall effect was modest, indicating that pain alone isn't a strong predictor of structural changes. Finally, baseline performance on the 20-meter walk test, particularly the number of steps, was predictive of OA progression, and baseline values were just as informative as those measured years later. Interestingly, changes in walking speed or pain over time didn't predict future progression, which suggests that early functional performance may be more important than changes observed over time. In conclusion, this study shows that simple, accessible measures such as sex, BMI, and a baseline functional test can help identify people at higher risk of faster OA progression. What this means is that clinicians could use these measures in early screening to guide prevention and monitoring without the need for expensive imaging or lab tests. Moreover, these findings highlight the importance of sex-specific risk models and early lifestyle interventions, especially weight management for those at higher risk. Lastly, I'd like to thank my mentor, Erica Secourt from Cornell University for her guidance and the Osteoarthritis Initiative for making their data publicly available. Thank you for listening. Hello, my name is Hello.
There should be another one. Oh, there we go. Okay. All right. Next up, we have Asha talking about bacterial pathogen survival in relation to produce. Sorry about that.
Should I start? Okay. Hello, my name is Asha Nay, and I'm from Alliance Academy of Innovation in Forsyth County, Georgia. And my mentor is Hasio Chandrani Chen from the University of Georgia in Athens, Georgia. And today I'll be presenting our work on bacterial pathogens on fresh produces. This is a systematic review about understanding bacterial pathogen survival on fresh produce and a review conducted of the plant colonization pathways and the pathogen's impacts on humans' health. It will include examples of some common gram-negative pathogens, including E. coli strain O157H7, the Salmonella strains, Listeria monocytogenes, and Campylobacter, as well as many others. Within the recent years, the consumption of fresh produce has increased significantly due to the raising awareness of health concerns and the corresponding shifts within people's diets. And while the consumption of fresh produce themselves do provide many benefits, a lot of the time they also provide transportation for bacterial pathogens. And while we consume this contaminated produce, the infections carry on into us humans and cause many foodborne illnesses and public health concerns to break out worldwide. There are many ways that these bacterium can enter the plants and contaminate them, both pre and post harvesting. Before harvesting, the bacteria can enter through the soil and contaminate manure or irrigation waters. The bacteria can also form biofilms, which allow them to survive in more extreme environments, making it harder to decontaminate the produce. Dirty tools and poor handling, as well as contaminated water after harvest, can contribute to the post-harvest contamination. However, for a few pathogens that can not survive long outside of their host, such as Campylobacter, post-harvest contaminations are a lot less common. After contaminating the produce's surface, some human inertic pathogens, such as E. coli and Salmonella, have the potential to modify the stomatal aperture, which can help the bacteria enter the plant's tissue. Factors such as pH levels and the moisture on the plant's leaves and surfaces also determine whether a bacterial infection will happen, and if it does get contaminated, determines and if it does get contaminated, it determines whether the pathogenic infection will persist. Factors such as the ambient humidity, UV amount, soil composition can immensely impact the pathogen's durability. For example, Campylobacter SPPs thrive in more moist areas with lower oxygen levels and more shade. Meanwhile, Listeria can grow in more colder temperatures, making it a threat in refrigerated goods and produces. Chemicals used within agriculture, such as pesticides or fertilizers, can also contain certain bacterial infections. Structural barriers, cubicle thicknesses, lignification, and stomatal densities also impact the plant's vulnerability. For example, romaine lettuces have thicker cubicles, causing it to have a lower chance of salmonella colonization than the other leafy green vegetables. Consumption of these contaminated produces can vary on factors like the type of pathogen, the infectious dose, and the individual's medical histories. E. coli strain O157H7 infections typically cause severe abdominal cramps and bloody diarrhea, which can then later progress to a life-threatening complication known as hemolytic uremic syndrome, which is most commonly found within children. Bacterium such as Listeria usually only affect pregnant women and can also cause miscarriages. Despite the many advancements within the understandings of these contaminations of the fresh produce, there's still a lot we do not know, like the mechanisms and environmental factors which allow the pathogens to internalize themselves into the plant tissues, and the role of biofilms in protecting the pathogens and how new technologies can influence these bacterial persistence within the plants. And even with these more technological advancements in new farming and sanitation practices, there are still many outbreaks and cases that happen today. And since the bacteria pathogens each have their own unique contamination and survival methods and required environments, prevention can be difficult, and it's essential that we be better understand these interactions and bridge the knowledge gaps in order to better protect the public health. Thank you. I would like to thank my mentor, Johnny Chen, for helping me and guiding me these past few weeks. And I'd also like to thank Harvard Open Bio Research Laboratory for providing me with this incredible opportunity.
Awesome. Yeah. Yeah, good research talking about obviously you can see it in the figure right there, I think is a good kind of simplification of it. But I also enjoyed the nuance that you kind of contrasted between the health benefits of eating fresh produce while allowing for contamination. Yeah, good work. All right, next up we have Avia talking about alterations in therapeutic potential of serotonergic systems.
Yeah, hi, my name is Ovia Mathi, and I go to Heritage High School. And my mentor is Christine Chow from the University of Minnesota. And our original work is based on investigating alterations and therapeutic potential of the serotonergic system during spinocerebral ataxia type 1 pathology. Now, to give an overview of our research, we're investigating the potential of serotonin-based therapies for SCA type 1, which is a neurodegenerative disorder marked by cerebellar degeneration and motor dysfunction. Though serotonin is traditionally known for mood and sleep, it also plays a crucial role in motor coordination. And there have been previous studies in other ataxias that have shown that enhancing serotonergic signaling improves motor symptoms, suggesting a possible therapeutic overlap with SCA1. Our study uses the ataxin mouse model, which mimics key features of human SCA1 pathology to assess changes to the serotonin receptors. Additionally, we're also using clinical data from the National Ataxia Foundation to further explore the correlation between serotonin medications and SCA1. Now I'm going to briefly introduce the cerebellum, serotonin, and SCA1. So the cerebellum is a major feature representative of vertebrae, and it integrates muscle activity to allow for motor coordination. Serotonin is known as a regulator for mood, reward, and sleep. And as mentioned above, it is also important in motor coordination and is distributed throughout the cerebellar circuitry. Early evidence showed that the serotonin system is altered in some ataxias. And since ataxia is characterized by lack of muscle control during voluntary movement, such as walking, swallowing, and speaking, there have been connections between ataxias and serotonin. Now, SCA1 is a autosomal dominant group of neurodegenerative disorders that affects the cerebellum and its associated pathways, and is classified by subtypes caused by trinucleotide repeats, typically CAG repeats, which encode for an abnormally long polyglutamine tract. SCA1 is one of the most well-characterized SCAs, and it is caused by CAG expansion in the ataxin 1 gene. And unfortunately, there are no FDA-approved cures or disease-modifying therapies for SCA1, which makes it a novel area of study. The methods used for this study were we used the female ataxin 1 mouse, which is a model of SCA1 that represents or replicates progressive cerebellar degeneration in mice. We used a total of 11 mice, so 2 wild type and 2 mutant per age groups, and we studied them at both 12 and 20 weeks. We took sagittal cerebellar slices for histological analysis, which was then stained to assess Purkinje cell health via calbindin and serotonin receptors expression. We also processed all the slices simultaneously, and imaging was performed using a Leica Stellaris microscope, and all the imaging analysis was blinded. We also used Fiji to analyze all the fluorescence images. We also used clinical data from the NAF, and we used patient data from the CRC database. The patient information included ataxia scores and medication history, which was then cross-referenced to assess whether serotonin treatments correlated with improved clinical outcomes. Now, when we looked at the results, the immunofluorescence imaging that we have below, so the 2 images at 12 weeks and 20 weeks, for both the wild type and ataxin 1 mice, exhibited a higher serotonin receptor signal intensity in the cerebellar layer as compared to the wild type mice. You can observe this through the enhancement of the red fluorescence in the SCA1 samples, which could indicate an upregulation of the serotonin receptor in the cerebellum of the mice with the disorder. The presence and location of Purkinje cells are also shown through the calbindin in blue, and the purple is the merged image, which shows the colocalization of increased serotonin receptor expression in these cells. This is also represented by the quantitative analysis in the bar graph shown below, which shows the serotonin receptor signal intensity in the cerebellar tissue to increase in SCA1 mice as compared to wild type mice at both 12 and 20 weeks, which indicates that there's an upregulation of the serotonin receptors, which is a consistent feature of disease progression in the SCA1 mice model, as observed in this study. We also condensed results from clinical data with the SARA score breakdown, which is the scale for assessment and rating of ataxia, and it's used to quantify the significance or the severity of the ataxia symptoms. And from all the patients that we observed, some of the patients receiving the serotonin-related medications actually experienced improvements in their ataxia scores, and one patient actually maintained the same score as displayed below. So to conclude our research, the quantitative immunofluorescence analysis revealed a consistent increase in serotonin receptor signal intensity in the cerebellar layer of the SCA1 mice as compared to the wild-type mice. And the clinical data of the SCA patients indicates that some individuals taking serotonin-related medications experienced improvements in their ataxia scores. So while these findings do support a possible therapeutic role for serotonin in SCA1, further research must be conducted to determine the therapeutic efficiency of this method of treatment. And lastly, I would like to acknowledge SRI OpenBio for providing me with this opportunity to conduct research, and also the SRI staff for their office hours and guidance. I would also like to thank Dr. Harry Orr from the University of Minnesota and the National Ataxia Foundation for their data. And lastly, I would like to thank my mentor, Christine Chow, for making this project a reality. Thank you.
Yeah, I really enjoyed that. I'll commend you on finding, this could be a stepping stone to future research, finding that possible therapeutic role for serotonin. So yeah, thank you for that. And I thought you had good figures displaying that. All right. This will be our last presenter, if I'm not mistaken. Um, Talking about machine learning models for early osteoarthritis, kind of like how there was somebody else presenting earlier. Anyway, Micah, I'll let you take it away.
Hello. Today I'll be discussing my comparative analysis of machine learning models in osteoarthritis, or OA diagnosis. OA is the most common type of arthritis, affecting over 300 million people globally with cartilage degeneration and joint pain. It begins when cartilage cells called chondrocytes shift into a catabolic state due to inflammation. This releases enzymes which lead to the breakdown of collagen and proteoglycans in cartilage. As the cartilage wears, osteophytes form, meaning that the bone gets denser and inflammation sets in. This triggers a damaging negative feedback loop of continual structural breakdown, and we can see the visual effects in the bottom left corner in Figure 1. OA not only causes mobility loss, but also a higher mortality and economic burden up to 0.5% GDP, and cases have also doubled from 1990 to 2021 and have continued to rise since COVID because of the sedentary lifestyles. Despite this, OA is often dismissed as typical aging, leading to delayed diagnosis and poor treatment, so early detection is essential. So in diagnosis, machine learning models take input data like imaging, clinical scores, and demographic info and generate outputs. There are classification outputs, which are categorical, like whether it is OA or not, and then regression models, which show a measured estimate, like how severe it is based on a numerical scale. Models have to undergo a training phase that may require labeled data, and testing evaluates how well the model works on new unseen data. OA also isn't caused by one thing, and there's different phenotypes or disease patterns, which affects how it progresses. Like post-traumatic involves injury, aging involves cell senescence and oxidative stress, and metabolic involves insulin resistance and inflammation. Other key factors include genetic disorder, genetics, gender, and social factors, and with around a 20% overlap in phenotypes, machine learning can help differentiate these. To compare different models, various model strategies like data sources or classification and validation techniques can be used. For example, models can be validated through holdout clinical cohorts using unseen patient data to be tested, and transfer learning is another method which tests a model trained with one data set on a completely different set. For example, a shoulder-trained model can be tested on knee imaging data. Imaging data is crucial, and MRIs offer detailed views of cartilage, but is costly and slow, whereas X-rays are more common and affordable. In Figure 2, we can see the radiography progression of the bone getting denser, especially from grade 0 to 4. Additionally, supervised, semi-supervised, and unsupervised are broader umbrellas for machine learning types. Supervised uses labeled data to learn its patterns. Unsupervised uses unlabeled data and allows the model to detect its own patterns, and semi is a mix. The breakdown's shown in Figure 3 at the bottom of methods, and further subtypes. Other elements of machine learning are feature engineering, which extracts the most important variables from raw data to lessen busyness, like principal component analysis reduces 3D joint structures into vectors, and statistical shape modeling only captures joint shape changes. And explainable AI increases transparency by revealing the thought process behind a decision, and different techniques prioritize different concerns. Overall, we can see how this fits together in figure 4, which is the top image and results showing a process map of how machine learning functions, with the input data and then the training phase in the gray box and classification in the colored boxes on the right. Table 1 shows all the models that were assessed against each other based on their individual methods and strengths, with the description column elaborating on specific techniques each used. And the ensemble of deep learning networks was the best choice, as it can grade based on is highly accurate and was also validated on. Other models either require large, expensive data, are weak in early stages, or lose spatial resolution because of feature engineering. And overall models that combine multiple data types and are validated across populations show the most promise. But either way, OA-focused machine learning has its limitations, such as an over-reliance on the same broad data sets, lack of external validation, black boxes, which are poor interpretability of reasoning, and underinterpretation of joints other than me. So going forward, more diverse data sets and explainable AI tools will be crucial for real-life application, and with machine learning, we have an opportunity to catch it earlier and personalize treatments. And I'd like to quickly thank my mentor and SRI for the guidance and resources throughout the program. Thank you.
Awesome. Yeah. I enjoyed that. I think models like this could definitely go hand in hand with other kind of racial or social disparities that we've talked about in other researches with that personalized treatment. So with that being said, I believe all of us have currently went. Yes, I should be at the end of this presentation. So nobody leave. We're going to head back into the main Zoom room. If anybody doesn't know how to do that, I can move you. But yeah, if everybody could click on. Uh, so actually, I might not know how to get back to the main room.
Just click leave room.
Is that what it is? You just click leave room, and we should be able to go back in there.
Yeah, leave breakout room after you press leave room.
All right. Yeah, I see everybody in there. Thank you so much.